Swayam Prabha. Digital India. Educated India.
Now, I will explain more about the contents of uniprotein. So, as I discussed earlier, it contains high annotation of the protein sequences. So, it is more information regarding a particular protein. So, what are the major aspects? What are the major contents of uniprotein database? So, first they give about the proteins, right, the names, origin and attributes and so on, and go with the ontologies, and then the next the sequence information, right. Then once we give this information, then they have to support this information by other means. So, the data they give in the primary data are supported with the bibliographic references, right, because it is very important to assess the reliability of the data as well as the correctness of the data. So, all the information they provide in the database, right, are supported with the bibliographic references where they obtain the original data, original information. Then they give the cross reference with lot of other databases. I will show some of the important databases, and they give the complete information regarding these particular entries. So, if you want to search data for any of these specific proteins, right, then I will show one example. So, hemoglobin B chain. What is the importance of hemoglobin B chain? Yeah, this is a transport protein, oxygen carrier protein, right. So, I put hemoglobin B chain. So, if you click the hemoglobin B chain and click on search, right, if you click here. So, this will show you all the entries related to the hemoglobin B chain. So, we saw here, okay, these are the various accession entries, right, these are the accession numbers, this entry name, and we have the protein names, right, we have the subunit beta, subunit alpha, subunit gamma, and these are gene names, right, and we have the organism, something from the human and something from this Mycobacterium tuberculosis. So, this is the length of the protein. So, it is 147 amino acid residues in this particular hemoglobin subunit beta. So, next they give the names, right, the synonymous names, what are the alternate names for this particular protein, right, then give the gene names, organism and so on. Then here we get the protein attributes, this 147 amino acid, its sequence status is complete because they got all the sequences, right, and it is known at the protein level. At the protein level they have the evidence, right, protein evidence at this protein level. Then you go with the general annotation, they try to incorporate or try to include most of the data available in the literature. So, what is the function of this particular protein? They transport oxygen, right, transport oxygen from lungs to the tissues. So, this is a major transport. So, then how many subunits or how many small subunits in this particular protein? Yeah, totally 4, right, because it has 2 alpha chains and 2 beta chains, right, you see the alpha 1, alpha 2, beta 1, beta 2. So, it is the tetramer, right, it is a heterotetramer, right, because 2 different chains, this way is hetero, there are 4 chains totally, so it is a heterotetramer, right. So, where is the found in tissues? It is in the red blood cells. So, whether any post-translational modifications in this particular protein. So, they mentioned was the glycation and different types of post-translational modifications, right, at this site. Mainly the acetylation on lyse 60, lyse 83 and lyse 145, right, and they give the proper references also. So, where they have the post-translational modifications. Then where this is involved in diseases, so they give a lot of information regarding disease. So, you can see they cause different diseases based on the mutations, right. So, they listed up several diseases where you can look all the details, right, from the uniprotein database. Maybe they give the sickle cell anemia, so by the mutation of 6 residue, right, glutamic acid to valine. So, they mention all the details about the diseases from this particular protein. So, now they give the ontologies, what are the various biological process, we say oxygen transport. So, any diversity of this coding region, so they have polymorphism, right, they have these changes upon mutations or any other ligands or any other small molecules they bind, right, with this particular protein. Yes. So, what are the different small molecules bind with this protein? Heme, iron, metals and pyruvate, right, these are the different ligands they bind with this protein. So, what are the functions and what are the post-translational modifications occurred in this particular protein. So, various types of PTMs, acetylation, glycation, glycoprotein, phosphoprotein as S nitrosylation, the various types of PTMs. Then we go with the ontology. So, there are different types of gene ontology widely used in the literature. One is a biological process, cellular component and molecular function. So, they give the different specific subclasses in this different gene ontological process. Then you go with these interactions, they have the interaction with other proteins. So, this is the database for the protein interactions, right, intact. So, they give the interaction with the other proteins, how they are interacting with the different other proteins. Then what are the different sites, what are the metal binding sites, what are different the binding sites, they give the information regarding the binding site of a particular protein, right. Now, the variants, there are various variants in this particular protein, right. So, they give the wild type residue, this is the residue which are really the original residue in the protein. This is the residue which are replaced. So, this is replaced. For example, valine is replaced to alanine, then it will happen to what is the major disease. So, here give you all the mutations from this is 2 and which type of diseases, and they give the references, right. They can have all the information. Essentially, if you see, UniProt is the unique resource which contain all the information regarding any particular protein, right. Now, we go with the next step. Now, till now they mentioned about the general information, right, what are the functions and what are the gene ontology, what are the binding sites, what are the PTM sites, all the information they give. Now, they go with the sequence level, right, the major aspect of this UniProt. So, UniProt contains not only the sequence, it contains the data for other different functions and the structure of a particular protein. So, here this is the sequence, there is how many residues in this protein, 14, 42, 43, 44, 45, 46, 147, 147 residues, right. So, you give the complete sequence, right, and here they give a secondary structure, because as you know this protein, right, this is the alpha helical protein, this is predominantly with alpha helices. I will discuss the secondary structure in the later classes. So, if you see the blue ones, they are mainly helices, right, and the green one strands, but here it does not have any strand, and we have some tands, okay, here and there you can see some tands, right. This is the secondary structure information regarding that particular protein. So, you can see the sequence in two different formats, either you can see this is the UniProt format, and also you can see the FASTA format. What is FASTA format? FASTA format is format which started with the greater than symbol, and here then you can see this is the command line, and here the sequence will start. So, this is a kind of format which we adopt in bioinformatics generally for the treating the sequences as well as for a large scale analysis. So, to separate two sequences they use this specific format. Then you can also see, if you see a sequence, any other sequences, right, any other organisms, they have similar to this sequence, right. So, they have various tools. If you see here, there are various tools available. One is BLAST, I will discuss the details in later classes, right. This will help you to see the proteins which are related with your original protein sequence. So, if you have your own sequence, if you click on this BLAST, this will give you what are the other sequences related with your own sequence. So, now if you have references, because this is the important part, because how they obtain the information, because they cannot get directly from from outside, right. So, they have to use from any reliable resources. So, one major resource, reliable resource, right. So, that is the published articles. Where shall we get this information? PubMed database, right. We discussed in the previous classes, right. So, PubMed provides the resource for all the published articles mainly in biology, right, and medicine. So, we have these different papers, you can get the information from PubMed, and they integrate all the data in the database, and wherever they collect the information from the literature, so they give the references, right, they give the references. To provide all the information, it is very time consuming. So, manual curation is very hard. This is the reason why the manually curated sequences are less compared with the computerly translated sequences. So, there they can collect the information by keyword searching, and they collect the data from the resources and put it as it is, right. They do not have to work on that, but for the reliability, so we need to do the manual curation. So, now I give the sequence databases, right. You can see the what is EMBL, GenBank and DDBJ, which sequence database? Nuclear sequence database, right. They give the complete sequence databases and the translated sequences, everything they provide the information, right. Then this is the 3D structures. So, this is the method how they obtain the data, right, and the resolution and the positions and so on, right. More details I will discuss in later classes, right. So, these are the protein interaction databases, MINT, Interact, intact and string. So, it will give you the your particular proteins, how this interact with other proteins. Then they give other proteomic databases and the post translational modification database and so on, right. Then also they provide genome annotations, organism specific database as well as the phylogenomic databases. This is the one part. First part is the general information, second part is the sequence and the structure and the functional information plus the links, and now they give the different type of annotations. They give the enzyme and pathways where they have this reactome and gene expression databases, right, and family domain database and as well as the other resources, for example the drug bank and other resources. Fine. Then they have the other relevant documents like human chromosome and other polymorphism, disease mutation and so on. Now, you are familiar with all the contents of UniProt database, right. When you have time, you look into the UniProt and take your own protein, and if you search and if you read, then you will be comfortable with understanding all the aspects of a particular protein. So, here the hemoglobin B chain, in the first part we give all the functional information regarding the general information, and second part with the protein sequence along with the secondary structure or the tertiary structure and the links to other databases, and the last part they give the enzymes and pathways and the interactions and so on. If you search with the hemoglobin B chain, for example, it will give you lot of data. So, you can for example, if you see this many data, and from that some of them are redundant. If you see the two sequences, they are sometimes 90% identity, sometimes 80% identity, right. For example, if you have these two sequences, what is identity? 100%, right. So, you can see the 100% identity. So, in this case, this UniProt provides an option to select the sequences with some level of redundancy. For example, if you want to reduce to 90%, that means if you two sequences, if the similarity is identity is less than 90%, you can get. With more than 90%, you will take one and discard this one. So, also you can get the reduced redundancy up to 50%. They will check the two sequences and see the identity. If it is more than 50%, they just keep one and discard others. How to reduce this redundancy, I will explain in the subsequent classes, right. So, you can get that. If you do this, here now currently we got 1243 results. When you look into this specific redundancy, right, now you can get 240 results. So, here we use the identity of 0.5. So, we reduced the data from 1243 to 240, because to do any analysis, it is very important to have the data from different ways, not to use the same several times, because you will introduce bias because of same sequence, right. So, in this case it is important to have a non-redundant data. So, here you can get with the two different cutoffs, right. For example, this is 100%, 90% and 50%, right. If you want to have other redundancies, we have to use other programs available in the literature. So, these are the entries. Now, you have 240 results. So, these are the different entries, right, obtained from this particular search. Now, you can also download the data, the various options to download the data, right. They have the various options. For example, you can see a tab delimited, and you can use Excel, you can download in FASTA, and you can download XML, and you can list the access numbers, right, as well as you can get the XML or RDF format. So, various formats you can download the data. So, if you give the FASTA format, right, so you give the FASTA format, this is the result. So, as we discussed earlier, the FASTA format is start with the greater than symbol, and this is the name and followed by the sequence. You can see from here to here this amino acid sequence, sequence number 1, and here again the greater than symbol started, this is sequence 1, right, and here this symbol started again. So, started the sequence 2. So, and this is a sequence. So, you can get all the 140 results based on the greater than symbol. You can understand, okay, first sequence starts from M and end with the RSF, second sequence started from MAN and ended with this AMRF. Then you can separate the data and you can use this data for the analysis. Now, I have two questions. First question is, obtain the sequences of transcription factors with less than 50% sequence identity. Transcription factors are DNA binding proteins, right, they have specific functions. So, to understand the features of these transcription factors sequences, so I would like to collect the sequence from the transcription factors and reduce the data with 50% sequence identity. This is question number 1. Question number 2, I am interested to understand the function of a particular protein. For example, this human mitochondrial beta viral membrane protein B.Tech. Two questions. So, to understand the first question, so how to obtain this information, how to obtain the transcription factors with less than 50% sequence identity. First you go to UniProt, right. First step is go to UniProt database and search for transcription factors. So, go to there and you can see the UniProt. So, you have to search with transcription factors. So, we search, this is the second step. So, when you search these transcription factors, what will happen? You will get a list of sequences which contain transcription factors in any field, because if you use a simple search, right, you can get the transcription factors which are matched with anywhere, or you can search in this, you can have the different options. If you want to get only the title, you can give the title, or any other mesh term you want to use it, you can use any mesh terms. So, with the transcription factors, how many data we get? So, more than 10,000 data, right. Now, if you use now, you will get more than that. So, now what is the question? So, we need the transcription factors and with less than 50% sequence identity. So, what to do with this? Okay, we have to choose the 50%, and if you click, you will get the data. So, to download the data, so there is an option called download here. Click to download, what will happen? It will ask for the option which format do you want. If you ask for the FASTA format, you will get the data in FASTA format, right. This is the one, and here this is the second one, and the third one, and so on. So, you get all the data in FASTA format, right. The first question is done. Now, you can use this for the analysis, right. What is the second question? Yeah, I want to see the amino acid sequence as well as some of the functions of this specific protein, because this protein is a recently published one. So, it has several functions, right. In the eukaryotic proteins, it is the first eukaryotic protein, right, in beta viral membrane protein. So, if you do this, right, just go to the UniProt, and if you search with this keyword mitochondrial beta viral membrane protein and human and B.Tech, right, then you will get this specific entries, and if you click on any of these things, right, you will get the data. This is a sequence. So, this is your sequence. If you want to get the FASTA format, you can click into FASTA format, and then you will get the sequence in the FASTA format. Now, in the UniProt, you can obtain the information on different perspectives. For example, if you are interested on any of the particular protein, you want to do that. So, you can do it and get the information. If you want to get the information regarding post-translational modification sites, right, if you give the codes, right, if you can get all the post-translational modification sites, or if you want to collect the sequences of a set of data, for example, DNA binding proteins or the RNA binding proteins or any information, right. If you give in the search, you search the collect, you will get the data, and it is also possible to get the data with any specific sequence redundancy. So, it is a unique resource. It contains a lot of information regarding protein sequences, right. So, this is the reason why several researchers, they are using the UniProt database in their research. So, now we will recollect again what are the various aspects we discussed in the class today. Primary structures, what is the primary structure? Specific arrangement of amino acid residues in a protein, right, like it has a main chain and it has a side chain. So, main chain, it is the same, right. What is the main chain? So, the amino sign, there is NH2, right, C alpha, C, N, C alpha, C, it goes on, right. Then you can put the COH. This is the amino terminal, N terminal, this is called the N terminal or amino terminal, the C terminal. So, you can see this is the information you will get, but C alpha is connected one side with the R group, another with the H, right. So, this is H, okay, this is R1, this is R2. So, now this is the main chain, this is chain is the same, only the side chains are different. So, you can get R1, you will get R2, R3 and so on. This can be same amino acid or different amino acid. So, this determines the sequence. So, here if you see this, what is R1 here in this sequence, this is M, R2 alanine, right, because M is here, right, and this is 1 2. So, this is how we get the sequence. This is the primary sequence. We know the link, we know the sequence arrangement, but we do not know anything else, right? We do not know the locations, right. So, right. So, now what is the primary resource for the protein sequences? PIR and the SwissProt, right. This is the earlier developed databases, right. Then they merge together to form UniProt database. UniProt is the universal protein knowledge base, right. Now, it is widely used in the literature. Now, what are the different contents of UniProt? Sequences, functions and the structural information and the various interactions and the pathways and assemblies, right, and supplemented with the original literature. Then what are the major applications of UniProt? Yeah, different functions and the different sequences, different distribution of the amino acids, different functions, right. We can use for the either specific sequence or any collectively sequences, right, and you can use it for the further applications, right. This UniProt is the unique resource for the protein sequence databases. Next class I will discuss about what are the various aspects you can derive from these amino acid sequences, that we will discuss in the later classes. Thanks for your kind attention.